I'm Erica Berg, Director and Senior Editor for Custom Publishing at Science, and I'll be moderating today's discussion. I'm very pleased to welcome today's panelists. We have David Lane, who's the chair of the Cancer Grand Challenges Scientific Committee. And then we have our inaugural cohort of Cancer Grand Challenges team leads. We have Greg Hannon, Cancer Research UK, Cambridge Institute. Give a little wave so everyone knows who I'm talking about. We have Josephine Bunch, Imperial College London and National Physics Laboratory. And we have Mike Stratton over there at Wellcome Sanger Institute, and Jelle Wesseling of Netherlands Cancer Institute. Before we get started, a couple of notes. We'll be taking questions at the end of this discussion from our audience here, as well as from our online viewers. So start thinking of your questions now, and online you can submit anytime by scrolling down in the right panel and clicking Join Event. And finally, thank you to Cancer Grand Challenges for sponsoring today's event. Now let's begin. While cancer remains a formidable challenge, with 1 in 6 deaths attributed to cancer globally, the past decade has seen leaps in our understanding of the basic science of cancer and treatment strategies. These advances stem from innovative strategies for pursuing the big questions in science, big questions in cancer research. This is epitomized by Cancer Grand Challenges. In 2017, Cancer Grand Challenges named its inaugural cohort of teams, each set against one of the most vexing challenges in cancer research at that time, which we'll hear about more shortly. So today, this challenge cycle is drawing to a close. So we're going to be looking back at how this unique team and challenge-based approach has changed cancer research and what it means for the future of cancer research. So first off, we're going to chat with David Lane. So thinking back to how this all got started, what was the vision and what distinguished the Cancer Grand Challenges approach from other funding strategies?
Yeah, thank you. I think the challenge is the sort of core of the whole idea. That is to pick up a problem that the community felt was a tremendous challenge to science, the science of cancer. And one of the things that's made the program, I think, coherent is always keeping the challenge in mind. In other words, why are we doing this research? What are we trying to do? And that then was the way that you could center a program, a global program. And it was a very bottom-up process. We really consulted the community, and we still do, to obtain those challenges. And then it was ambitious. We wanted big grants because we were providing big money. So the whole idea would be that it would be a way that you could bring together a really big team. And I remember one of the scientists saying it was like being in charge of a new university, and you could just hire the faculty you wanted because you could get everybody that you wanted to work together to come and work with you. And I think so that was very important. So the kind of global nature and the scale and scope required to tackle the challenges. And then I think the challenge selection process was very important, and I think it turned out to be extremely successful, as we'll hear from the people who've run these programs. So the first challenge we set was all around unusual mutation patterns, and this was really because we were beginning to realize that the technology around DNA sequencing, the ability to look for particular patterns of mutation was now coming into fruition. And so that was a very exciting challenge. We had examples of natural plant products, for example, that are highly mutagenic, and we're beginning to see those kinds of effects. And then the other one is was a very fundamental challenge, which was around lethal versus non-lethal. And as you'll hear, this one's centered in the end around breast cancer and the condition of ductal carcinoma in situ, and this appallingly difficult clinical decision as to what should happen when that kind of diagnosis is made, with a sense that, you know, over-treatment was happening a great deal, similar to the problem we've seen in prostate cancer. So how do you distinguish a lethal cancer that needs to be treated from a non-lethal cancer that doesn't? And then the third challenge, to which we got a tremendous response, and in the end selected 2 teams, was around 3D tumor mapping. And again, it was this sense of technology breakthrough, that new methods were coming that would allow you to characterize the components of a tumor. And this, you know, amazing early studies just showing that how complex the tumor microenvironment was, how many components there were, the beginning of, you know, the enormous impact of immune oncology, realizing that the immune components in the tumor are extremely important, and that there were new technologies coming. So those challenges are, you know, what has set us all on this path, and I think the example of what's been achieved was incredibly inspiring and taught us a lot about how to work with a team. So all of these people know me pretty well, and you know, we've had a lot of interactions, because this has been, as I said in the earlier introduction, a much more interactive way of doing science, where the funders are engaged and supporting and trying to help the teams all the way through, rather than a classic response mode funding where the decision is made to fund and then basically you're inspected for good behavior. This was much more proactive, where we're actually trying to help people to be successful. So those are the, you know, kind of background to the program, and it's been incredibly exciting to be involved with it.
Great. Thank you so much for setting the stage so wonderfully. So now we're going to hear from each of our team leads about why did you want to address the challenge that you addressed, and how did you pull the team together that allowed you to address that challenge? And Josephine, your challenge was 3D tumor mapping, one of the leads of 3D tumor mapping. So let's start with you.
Why did I want to address it? This isn't a field that I'd worked in before this program.
Oh.
In fact, I heard about the Grand Challenge opportunity on Radio 4. It was picked up by BBC Radio 4's Today programme. It's not an area I'd ever worked in, and it was presented as a measurement challenge: find a way of mapping tumours at a molecular and cellular level, as a call to arms, a global call to arms. And I'm based at the UK's National Measurement Institute, the National Physical Laboratory, and we have a collection of modalities which are useful for producing spatially resolved maps and images and spectroscopic data of wide-ranging molecules. And we felt very strongly that we could provide useful measurements to this challenge. In terms of bringing the team together, that was partly organic. As a starting point, you ring a person you know, you ask them if they think this is exciting, they say they think it's exciting. I'm not sure that they want to fund you, but you start working on a team to bring that together. And from that, we realized that with a scale of the funding available, you didn't just have to produce a proposal based on a very small team who already worked together. You could think about where the gaps in your approach were and who might be the best minds to fill that. You could bring together not practitioners, but in fact inventors of what might be the most exciting technologies. And you can bring together the pioneers of the people, in our case, the people who are pushing the boundaries of thinking about metabolism and its measurement in cancer, and who would make the biggest use of spatially resolved measurements. In our project, we didn't focus on a particular disease. In fact, we brought together leading researchers exploring breast cancer, glioblastoma brain cancers, colon cancer. We were so fortunate to be able to bring in a partner, AstraZeneca, which really helped us test this in a whole range of preclinical scenarios, and really push the boundaries of how we thought some of these modalities might be adopted.
Great. Greg, how about our other 3D tumor mapper? Same question. Why did you want to address this challenge, and how did you pull your team together?
Yeah, so I guess in some ways, so I moved to the UK 10 years ago and was in New York before that. And in some ways, the interest in the 3D structure of tumors came out of collaborations that I was doing with neuroscientists, trying to map neuronal connections. And one of the tools that they used was this serial two-photon tomography microscope, where they could take, entire mouse brains and produce, subcellular resolution 3D images. And, as we were working with them, I kind of realized that, people didn't really know what the spatial organization of tumors looked like, except from, 2D sections, sampling, histopathology slides, et cetera. And so when we moved, it still, probably shouldn't say this because CRUK spent a fair amount of money on the instrument. It was still a bit of a hobby project. And, we were making progress, looking for things like disseminated tumor cells at secondary sites. And then the Grand Challenge came out, and I was actually quite surprised to see the 3D tumor mapping one, because I went to one of the kind of final bottom-up meetings, and it wasn't mentioned, and I think it arose from the external blue ribbon panel. And I thought, well, what's interesting about this is, so we had this kind of idea that we should try to understand what the structure of tumors looked like, but here was a way to really power it, and not just in an imaging way, but to hang molecular measurements underneath those images and really, produce these, very detailed cellular resolution maps and try to understand whether organization mattered. And so I think one of the key things about the Grand Challenge is this kind of shortlisting period where they give you a little bit of money to, kind of form your team and form your strategy from, what is the initial idea. And so that was great because, we could start to think about, well, what kind of measurements do we want to make? I've heard Josie say she wants to measure everything. And I think what's true about these two teams is we measure very complementary sets of molecules for the most part. And then to think, well, how are we going to do that with emerging, nascent, sub-nascent technologies, as it turned out in many ways. And so we looked for the inventors of those, much like you did. And we formed kind of a hub and spoke model where, we brought together a series of people in Cambridge from lots of different disciplines, including astronomy, chemistry, mathematics, computational biology, cancer biology. And, sort of, that was the kind of the biology hub in some ways, but then out to spokes, which not exclusively, but were people who were really pushing the edge of these technological advancements.
Great, thank you. Jelle?
Yeah, the grand challenge for us was really, optimize management of a lesion that was framed as stage 0 breast cancer. So that comes with a lot of damage, so to speak, for a lot of women confronted with this diagnosis. And personally, I was really annoyed, so to speak, in 2010 or 2011, that for a certain lesion I saw under the microscope as a pathologist, that the decision was mastectomy or breast conserving surgery plus radiation. And then this call came along, and to really address this issue, you need a multidisciplinary team of the best experts worldwide. And there's no funding scheme, there was no funding scheme, I have to say, that could, help you in doing so. And this was so timely that we stepped into this opportunity. And we're building a team from clinicians all the way to computational biologists, epidemiologists, etc., that allowed us to collect sample and clinical data series across the globe. And that's a challenge in its own. And David referred to this morning already. But we did, and that has yielded so much, I would say, impact already in the field that, for example, in the Netherlands, for low-risk DCS, about 80% of the women, they opt for active surveillance and not for treatment. So timely, global, multidisciplinary, with the best experts in the world, and impacting real clinical practice all the way from biology, because that's also a disadvantage of regular pathology. It's a 2D snapshot, and therefore we really focused also on the dynamics of the lesion, intravital microscopy, preclinical models. What can we learn from it for the clinical assessment of a risk, etc. So yeah, for us it's obvious the opportunity that we have taken to address this.
That's great, so timely. And Mike, you were on the mutagen challenge. How did it go for you? Why did you tackle it?
So we had been working for a number of years on mutational patterns, which was the words that were used, I think, in the challenge, so-called mutational signatures, working to develop the conceptual framework, the computational ways of extracting mutational signatures, demonstrating the repertoire of these signatures, which basically represent mutational processes across the range of human cancers. And the advent of this challenge offered the opportunity, and it was very broadly put, very non-directive, of applying that framework to a major specific question in cancer. And I was contacted when the challenge was publicized by Paul Brennan at the International Agency for Research in Cancer. And we had some discussions together. And the insight that Paul brought, which I think framed the centerpiece of the Mutagraphs Grand Challenge, is that although there has been tremendous success through conventional epidemiology in identifying causes of cancer, form of tobacco, ultraviolet light, the various infectious agents we've been hearing about, there was strong evidence that there were still causes of cancer that were unknown to us, and that to a certain extent epidemiology was having trouble identifying them. So an alternative approach could be helpful. And the virtue of applying an approach using mutational signatures, in other words, finding mutations in cancer genomes, finding unknown patterns of somatic mutations, is that it didn't require us to make any particular hypotheses as to what those underlying causes were. We could just systematically look through cancer genomes, well-chosen sets of cancer genomes, to see if we could find unknown patterns which would reflect unknown underlying causes of cancer. So that was the center of the way we thought about it. But in order then to build a team to deliver that, essentially what we were doing was establishing a major strategic alliance between cancer epidemiology and cancer genomics, a strategic alliance at a scale at which it hadn't been done before. And to do that, we needed people, Paul and IARC, who had the expertise in epidemiology, had the network that comes from the International Agency for Research on Cancer for identifying cancers, patients around the world. So there was that part of the strategic alliance. And then the genomics, yes, sure, for generating the data, but for seeing the mutational signatures, the computational approaches that could be, that needed to be applied in order to extract that information. And then alongside it, to inform those patterns that we might find in the naturally occurring cancers, experimental approaches using candidate carcinogens, either in mice or in cells in vitro, that would produce patterns that we could match against those that we found in naturally occurring cancers. And the final part of it was that this was still at a relatively early stage of the period in which collectively we were all beginning to explore somatic mutations in normal tissues. Tens of thousands by then of cancer genomes had been sequenced, and that had become a relatively routine thing to do. But for technical reasons, it had been much more difficult to explore somatic mutations in normal tissues and in normal cells. But those technologies, those approaches were just being delivered. And of course, when carcinogens act, they act on normal tissues. They don't act on the final cancer, they act on normal tissues. So we needed to explore what was a a landscape in the dark, really, at that time, of somatic mutations in normal cells. And so it was with those principles in mind that we assembled the team.
Great, thank you. All right, so the challenges have been accepted, the teams have been formed. Now we're going to fast forward a bit and say, and the next question is, so how has your team changed the field, and do you think that your challenge is still a challenge, or is it done? Let's start with you, Greg.
Well, I mean, I remember kind of early on, sort of talking to people about this and talking about how, all biology happens in 3 dimensions, from development to cancer. And that we needed ways. And I think, nobody used the words spatial biology when we started, because it didn't really exist. There were measurement techniques that were starting to emerge, but it wasn't really a field. And I think what's really happened over the course of the last 7-ish years is this has gone from something that, was almost in people's imaginations to, a true field of spatial biology. There's been an absolute explosion of approaches, quite a number of those coming from members of our team, but also from elsewhere. There's now commercial solutions. I think only one existed when we started, and it was still fairly early stage. So I think what we've helped to do is to establish this entirely new field, a new way that people now think about these kinds of problems. Well, the other thing I'd say that has emerged, from, we'll talk tomorrow about the different biological findings, but just the fact that spatial organization is important, right? That was a hypothesis. It was a bet that was made. And I think that was also one of the key findings of the team. Now, is it still a challenge? I would say it is, because I say we're coming to a point where, someone can buy an instrument and make a measurement. Where I think what we've done has been important, and where I think the field still has to go in a kind of more democratized way, is taking measurements of different kinds of molecules and putting them together in a spatial framework. And I think we still don't quite have a technology that will make those multimodal measurements on a single piece of tissue. We largely infer their overlap. And I think, that's one aspect as to why it's still a challenge. And I think the second aspect as to why I think it's still a challenge is this whole democratization idea is, these technologies need to be disseminated and deployed across, many different tumor types, sample cohorts, et cetera. And I think we're just really on the cusp of that.
Thanks. Josephine, you are on a similar challenge, so what are your thoughts on that?
I broadly agree, with the exception that you can't do it on a single section. I think that even that's catching us up. I mean, you're absolutely right that we're witnessing a revolution, not just continued evolution of technologies, but a genuine appetite for these measurements that has gone hand in hand with the pace of their development. I like to think that many people in this room have played a part in that. I think they will play an ongoing part in that as their results start shaping the ideas and ambitions of other people who might seek to go on and use these measurements. There's still a long way to go. I mean, I think probably even up to a couple of years ago, I would have said that the next part was the ongoing performance issues, the classical metrology of how do you translate these techniques into things which are usable by others, sufficiently simplistic, repeatable, etc. But actually what we've learned over our grand challenge for 7 years of research is that while according to classical calculations of repeatability, many of our measurements are not repeatable, interestingly, by the end of the program, our findings are somewhat reproducible. And that gives us, from a measurement science point of view, some very interesting further avenues to explore as we think about how to support wider use of these and how to maintain equivalence as these start being used more broadly in preclinical and clinical research.
Jelle?
Well, there are a few things or more things to mention, but I focus now on a few very, I would say, intriguing findings from the preclinical models that can translate to the human setting as well. So in the preclinical models, we combine the models as generated by, for example, Jos Jonker's lab, together with the intravital imaging as done by Jacob van Reene. And that's one example of an investigator that feed it in later on. So really illustrates the flexibility of the cancer grand challenge, which is of great value. And then we discover that the growth pattern, so I will tell more about the details later on Friday, really relates to the risk of progression to invasive breast cancer. And how can we translate that then to the human setting in a way that we can assess this in the clinical setting? So, and what drives these growth patterns? And we're not there yet with the answers, but again, we would like to understand the biology of the lesion and then see what the readout is in the human setting when you see this 2D, 3 micrometer thin section in front of you. That is, I think, there we made major steps. Also, the adipocytes that really play a role in the risk of, pathologists never look at adipocytes in mammary tissue. It's not interesting, but they play a role in the risk of progression. What's going on there? And then discovering that the COX-2 expression, for example, in your DCS lesion and your adipocytes somehow are, interacting with each other. Well, that was quite an unexpected finding that we're validating right now. Most importantly, I think, well, there, I can list another 4 or 5 findings that are quite striking to us, but that also that we knew really almost nothing about DCS before we started. And I think it's a great gain that we catched up, so to speak, with the knowledge that was there already for invasive breast cancer.
And Mike, change the field, challenge, still a challenge, not a challenge anymore?
Well, I think the first thing is that the Mutagraphs program has demonstrated what is possible. In colorectal and clear cell renal cancer, for each of them, 1,000 cancers were collected, and for each of those in 11 countries on multiple continents around the world. And those collections, with all the ramifications of regulation, the operational aspects, recruitment, consent, bringing those samples, making DNA, putting them into a pipeline and processing the data, that's all a major challenge to do on an international scale, and that was achieved and will be the foundation of going to a greater scale in the future. So that's the first thing. The second thing is that it is clear from the data that emerge, for example, from the renal cancer study and the colorectal study, that there are major mutagenic exposures that are going on. And perhaps compared to some of our best known mutagenic exposures, such as smoking or ultraviolet light, these are silent and unconscious. The people who are being exposed don't know they're being exposed. The third thing is that some of these exposures are affecting millions of people, and some of them undoubtedly tens of millions of people. These are still exposures occurring around the world, and we do not know the full extent of them as yet. Some of them are kind of geographically very limited, and some of them may be quite ubiquitous. And so in doing these studies, we have really established that sort of geographical, that sense of what is going on in the world. Some of those exposures we do know the cause of. David alluded to one, the extracts of aristolochic plants, genus of aristolochic plants, so-called aristolochic acids. These are highly mutagenic chemicals, compounds which are known to be carcinogenic, these must be affecting tens of millions of people. We see our epicenter of this in southeastern Europe, in Romania and Serbia. We also found it in Thailand, we found it in Singapore, we found it in India. This is from relatively limited sampling of 11 countries. So that exposure is really very widespread. Okay, for that one, we probably do know the cause, although we don't know how that exposure is getting into people for the most part. But for others, in renal cancer, we found a mutational signature, a pattern of mutations, which was present only in Japan. So far, we cannot find it anywhere else. It appears to be a localized, and it has all the features of a mutagenic exposure, so I won't go into, but they are really those features are associated with mutagenic exposures such as tobacco and aristolochic acid, and it has all those sorts of features, but it's restricted to Japan, and we do not have, and our Japanese collaborators do not have any idea what this is due to.
Well, that is terrifying.
And then there are the hints of other types of exposures which are actually quite ubiquitous. They have the indications that they're exposures, but they're ubiquitous across many countries, but the extent, the intensity of the exposure differs between countries. So this is what I think we have begun to uncover, this landscape, and it defines a number of further issues to address. In finding these exposures, we have looked at a limited number of countries. It is quite possible that we have missed others that are active at great scale. We need to define their geographical boundaries, and there is work in progress to do exactly that. Two key questions that arise for all these is, one, what is causing them? Well, for the Japanese exposure, we just don't know. And what we can find is a mutagenic exposure. What to be proven is the extent to which they're contributing to cancer incidence, and that's a whole series of studies that needs to follow on.
So your teams have made tremendous contributions to the cancer research literature over the course of these grand challenges. So what I'm wondering is, did you uncover anything that was particularly surprising to you, something you were not expecting? And David, I would be interested in your thoughts on this as well.
Yeah, I mean, I found everything surprising, to be honest. I think, you know, in each of these programs, there were unexpected discoveries. So I think in the breast cancer example, the level of genetic alteration in very early lesions was very surprising, and it's very clear. So you have cells with aberrant mitoses, with chromosome translocations, mutations, and yet these are not going to lead to cancer. So it's a kind of pretty strong rewire of what we think about tumor suppression and how it works. Is it immunological? Is it structural? The kind of tools that our mappers have developed are going to be very important, and Jelle hinted at that there's, things like growth pattern are going to be very important in understanding. So that, to me was, a big surprise. I think Mike emphasized one aspect of his work, but there's another aspect that's led to the prominent program, which is the realization that in some types of cancer, a regional difference is not directly related to mutational pattern and appears to be related to tumor promotion. So what has been coming more apparent is this earlier work which showed that you could have a mutant cell that would sit there doing nothing until another substance, a non-mutagenic substance, changed the environment in which that cell existed, and then it would be promoted. And I think this, again, is very kind of central to our understanding of tumor latency. And I think, what emerged from what both Josephine and Greg did was this, just absolute proof of the heterogeneity of cancers, both at the physical, large protein scale, but also at the details of metabolism, and the absolute proof of things that are sort of people had thought but hadn't shown. So this amazing formation of blood vessels that happens in tumors from cells that are not blood vessels. So a completely different understanding of how that develops. And then in Josephine's case, I think one of the remarkable things was the immediate uptake by AstraZeneca, the realization that these measurements could help very, very quickly in understanding how drugs were working in a tissue, that they could see very clear evidence of metabolic change and measure it very accurately. So all of these, brought tremendous kind of unexpected insights. But my whole view of cancer has been changed by the combination of these things. So it's a very different vision. Maybe I was naive before, but I just see things differently now. I see the mutational spectra as a part of a very much more complex process, one that involves tissue structure, the immune system, all sorts of feedback loops, metabolic alterations that I didn't see when I read Weinberg. So I think it's and the other thing for me has been just to see the power of team working, the ability to actually progress in this way, and also emerging very strongly is actually the crosstalk between the different teams. So I was always optimistic. I mean, I've been a great enthusiast for this program right from the start, but I think it's exceeded my expectations in many ways. And I suppose my final comment is actually the community. I mean, I think this group of people working in this way, team working, global working, has changed the kind of the atmosphere. It's created a very innovative environment for people. And in a way, Josephine represents that. She comes in from a career built around physical measurements of things, and now she's a cancer biologist. She forgive you for saying so, but I mean, that is a kind of transformative and very inspiring kind of process for me. So that's my answer to your question.
Hello?
Yeah, so related to what David is saying, we were not so much surprised that the genetic alterations present in early DCS lesions were quite similar or very similar to what you see in invasive breast cancer, but that we then found quite a different RNA sequencing signature in the high versus the low risk DCS lesions. And why is that? Because it's not so much driven by the, I would say, simple sequence of the DNA itself. Is it the extracellular extrachromosomal DNA? Is it something in the surrounding? Thus far, we couldn't find a hint in the immune microenvironment. Is it in the adipocytes, etc.? So, unfortunately, each grand challenge delivers a new grand challenge. Sorry.
Mike, I was wondering if you could talk a little bit more on, I think, what David was alluding to, the promotional carcinogen challenge and how that came about.
Well, the first cancer type that we looked at was esophageal squamous carcinoma. And this is quite a remarkable cancer in the variation in its international incidence rates. It has areas of very high incidence in East Africa and a belt across Central Asia, and very low incidence rates in sort of developed countries. And there have been a number of hypotheses made as to what was the underlying cause of this, but none of them appear to account for these very big differences in cancer incidence. So, through the offices of the International Agency of Research on Cancer, about 600 esophageal squamous carcinomas were collected from 8 countries, and these were sequenced. Given the differences in incidence rate, we were expecting to see a new mutational signature, different patterns. What we saw across the 8 countries was almost the same pattern of mutations in each one. Almost the same pattern of mutations, almost the same set of mutation burdens. We really couldn't find anything that was accounting for these differences in incidence. So, in the end, carcinogens need to have their effect on normal tissues in one way or another. One way is to increase the mutation burden, and that will result in an increase in mutation load and then increased numbers of driver mutations, which may cause cells to become cancers. But the alternative hypothesis is that actually there are agents that change microenvironmental selection pressures. And what they do is they act on cells that already have driver mutations, encouraging them to grow faster. And that leads to more driver mutations in the normal tissues. That possibility fitted very well and sort of came out more or less at the same time within mutagraphs as the work that Alan Balmain was doing in rodent models. Because there, Alan had taken about 20 putative human carcinogens, and mice had been treated with these carcinogens, and they had developed tumors. So the carcinogens, they were working as carcinogens. But when Alan, as part of mutagraphs, sequenced the tumors, he could only find, I think, elevated mutation loads, mutational signatures for 3 of those carcinogens. The rest of them had the mutation loads and signatures that you'd find in a spontaneous mouse tumor. So this was coincidentally, convergently, an indication that many of these putative human carcinogens were actually, what name are we going to give them? Promotional carcinogens. That's a word that comes from the work that was done in rodent systems 50 years ago, when Alan was in his early years as a scientist. But you know, a huge, massive, really long tradition of work in this area. We can give them another name. You know, cancer is about somatic evolution in cell populations. So one set of carcinogens are mutagenic, the alternative set you can call selectogenic carcinogens. And that's what, at least one hypothesis for what is happening in esophageal squamous carcinoma and is the basis of the prominent grand challenge.
Greg, I was wondering if you could share, how did Cancer Grand Challenges enable you to do science different than how you were doing science before?
Well, I mean, I think one of the ways that, well, I would say 2 things, right? So one of the ways that we approached this was not, I mean, again, I talked about the hub and spoke model, but in the Cambridge hub, what we didn't do was distribute the funding amongst, 7 individual labs or 8, depending on the timing of the number of PIs we had in Cambridge. We appointed one later. What we actually did was formed a completely new research group with its own research space, co-supervised by that group of investigators. And in that space, we sat all of those different disciplines. And so everybody sat next to each other, and the kind of different perspectives that they brought were shared because of that shared geography. I think in a way that wouldn't have happened if we had N standard research grants and just formed a collaboration. And I think it would have been very hard to do that without the kind of muscle of the Grand Challenge. Research space is always at a premium, I think especially so in Cambridge, but to have the kind of cachet to say, I want to effectively start a new lab that works in this way. And so I think we couldn't have done that. The other thing, I think Mike or Jelle mentioned the flexibility. We did something in our original application that would never have worked, I think, anywhere else, which is that we left a fairly substantial pot of money completely unallocated. And the reasoning behind that was that, also argued that, a grand challenge takes more than 5 years, but, we don't know what the world's going to look like, particularly in such a fast-moving field. And so the combination of those 2 things gave us both that cross-fertilization, but also the flexibility to bring in additional people, which we did, as new technologies emerged or as it became clear that we needed different sets of expertise to progress what we were doing. And I think what's changed for me, and I can mention that there is an astronomer in the audience, Nick is out there somewhere, who has attended every single Grand Challenge meeting. And I think, it's like bringing new people into the field, but I think one of the surprising things to me, and it really came from creating this shared research space, is how things that we view as very different disciplines can interact in a really surprising way. And I think our collaboration will certainly persist after the Grand Challenge ends.
And we're getting close to question time, so I just want to have one more super quick answer for this last question. But what does the future look like for your field and for cancer research? Rapid fire. Go, Josephine.
Oh gosh, I'm not the right person, but I hope that for many people they will include spatially resolved metabolism in each of their studies, alongside, I think, there's already a plethora of tools for spatially resolved protein measurements, and more recently of spatially resolved transcriptomics. But we really do have the opportunity to perform genuine multi-omics on single sections now, and I hope many of you will.
Same thing.
Very efficient. Thank
Yeah.
you. Moving on.
Okay, for us, it's really, conquering overtreatment. And that's very important. And for low risk, low event rate diseases, we talk a lot about stage 3, stage 4 disease. But at the other side of the coin of, early detection, we suffer from high sensitivities and specificities that are way too low. And when we understand the biology much better than we do now, we can select out the patients that really need interventions from the ones that do not. I think that's a major challenge. Also, when it comes to keeping healthcare affordable, and we learn a lot, you need to build large international data series, sample series, image series, etc. That's the only way to tackle the problem.
I think Mutagraphs has established the operational feasibility and scientific worth of this field of mutational epidemiology, that collaboration between epidemiology and genomics. And I think it will be but a pilot for the much bigger studies to be done over the next 20 years.
Yeah. It's no secret that Mike usually asks me for another 100 million when we meet. So. For me,
Sure.
I guess,
Yeah, please.
I think that what's next for Grand Challenge is, I think, to grow the program. I'm personally very convinced this is a very good way to fund science, and I think, with great respect to all of my colleagues at the NCI and CRUK, if you look at the fraction of their research spending that goes here, it's relatively small. And I think what we need to do is engage with the world community, the philanthropic community, to help people understand that this is a very good way to do science, to deal with the problems that we have. And it was interesting that, I'll try not to rabbit on, but it was very interesting, this process where after we did the initial expression of interest, people are given money to write the grant. So that actually, I think, is a very interesting thing to do. It doesn't normally happen, and it does give people the opportunity to pull their teams together. You've got to fly and look in somebody's, to America, and look in somebody's eye, or vice versa, to be sure you're going to work together for the next 7 years. So that particular thing that was brought in, I think, has been very, very effective. And actually, some of the unfunded teams have come up to me and said even that was incredibly helpful. Got us all working together. So, yeah.
Great, thank you. So questions around the room. There should be microphone somewhere. I'll just start with a question from online while we get the microphone situation in the room. What piece of advice do you wish you had been given when you started your Cancer Grand Challenges Award? Who's got something?
I could do a real quick one.
Okay.
I think the one thing that we really learned was to make sure that as quickly as you can, you transfer ownership to the more junior investigators in the group. Because, science is a ground-up enterprise. And I think really bringing the students and postdocs to the front and, allowing them to shape the science has been really important. And I wish we'd done it a couple years earlier.
Yeah, I think it's really pivotal that you, and it's a cliche, but that you share the very same end goal, because then you can go back in your discussions, your conversations, because everywhere around the world, also in teams, some egos are bigger than others. Could be a song, but I think it's important to, jump beyond your shadow, as you say in Dutch, and really go for that end goal. That's really important.
Great. Do we have a question from the room? Hello.
This is mainly for Mike. So I'm a stem cell biologist and have isolated the blood-forming stem cell and looked at both humans and mice. And the amazing thing about stem cells is in the blood-forming stem cell, they're just 1 in 100,000 cells in the bone marrow. Yet they're the only self-renewing cells. Self-renewal means you can maintain a lineage for the life of that animal. You could transplant it for another animal. All of the other cells, you transplant them and they just have a short lifespan to make the tissue. So that says, if you're going to have more than one mutation necessary to make a cancer, and with the, I'm guessing, the low probability of mutations, it means it should be growing as a pre-leukemia, for example, in the stem cell. And we showed that in AML over and over and over again. What was unexpected was that the order of mutations in terms of biological functions are not random. So the first one that comes up that makes a driver mutation is something that's involved with methylcytosine or demethylating it so that you can open or close chromatin. They're loss of function mutations. The cell now has trouble getting out of the stem cell state, but it still divides. Then there's a couple of others. There's usually 4 driver mutations. The last one, which the hematologist told me would be the first one, were the known oncogenes, HRAS, KRAS, beta-catenin activation, FLT3 internal tandem repeat. All the last. So there's no logic that I can think of that would tell you that order ahead of time. And second, since mutations are random as to the cell they happen in, the time they happen in, does this really mean if there are 4 mutations and there's a particular order, you don't have 256 ways to make it, but you have 1 or 2 ways? So, Mike, how could it do?
Yeah, I could kick that in, let's discuss afterwards, but I suppose I would agree with your final comment there. I don't think it necessarily applies to all cancers, that there is a kind of rather rigid order in which the mutations occur. For some other cancers, there's greater flexibility in which the order in which they occur. But yes, I would agree with your conclusion that if there is that order, then there's a sort of narrower corridor through which a cell has to navigate its way in order to get to being a cancer finally. It still will be helped to do that if its mutation rate is increased, and the exposures will help it do that. Yeah.
We're testing one other cancer now. That's what we're doing with the grand challenges. It's the only other one that we really have the stem cell for. That's glioblastoma and neural stem cells. So it'll be interesting to see if every cancer follows the same route. I'll just say we never saw a de-differentiation. I was taught by my medical school professors, oh, this is a de-differentiated cancer. But we've never seen a single step of de-differentiation in normal or neoplastic cells.
Great, thank you.
Yeah.
Let's take another one from online in the last few minutes. What was the impact of patient advocates on your team's work? Jelle, you had some?
That was huge from the very beginning. So they were involved already with writing the letter of intent Yeah, we addressed, or we're trying to address, a real societal and clinical problem, all the way from bottom up, from the patient perspective, but also from the biology perspective. And they always asked us the question: how can we explain this to our fellow patients or to the community? So that was not always easy, because sometimes it's hard to explain, but it was really useful, and I think it added great benefit to steering the direction you were going. So it was crucial.
Could you really quick follow-up?
Please.
I think the one thing that my long experience with patient advocates has taught me is that, they'll hold your feet to the fire. We're scientists, we want to do things that are interesting. What they'll do is make sure that we do things that are important.
Yeah, and I think that's a great way to close this out. I just want to thank all of our panelists again, and Cancer Grand Challenges for bringing us together to have this fantastic conversation. Thank you so much.